Thanks. This was a really great start to the conference. I've been enjoying it. We're going to have a 5-minute break, and I think we were supposed to come back at 4:30, but we're a couple minutes late. Should we give the full 5 minutes, perhaps? Okay, so 4:33, come back at 4:33 to start with our students' lightning presentations. In the meantime, if you're hanging around, we're going to share a slideshow that are elementary school student posters, submissions to the North Carolina Soil and Water Conservation District's annual poster contest. And their theme was, we all live in a watershed this year. So we thought that was a great partnership for this conference. So I'm gonna go ahead and start that presentation. You don't have to sit and watch it unless you want to, but it's here for you. And Anna is sharing out a link where you can find that if you want to watch it later on your own time. So enjoy your 5-minute break.
All right, I hear it.
All right.
Welcome back, everybody. That was a quick 5-minute break, wasn't it? We are now ready for our wonderful student lightning talk presentations. And so we've got a wonderful set of talks here for you. And we're going to kick it off by inviting Risa Sayre from UNC Chapel Hill to give her presentation on the quest for average water, determining representative surface water concentrations for chemical risk assessment prioritization. So with that, Risa, take it away.
Thanks for that introduction, John. I'm going to invite you to follow a few of my steps on my quest for average water. I have done this work as part of my PhD at Chapel Hill, but I also work at the U.S. Environmental Protection Agency, which is pictured behind me. But I want to mention that this presentation doesn't necessarily reflect USA policy. So why do I want to find average water? What does that even mean? I want to find average water for risk prioritization. I think I need to minimize this here. So there are tens of thousands of chemicals in commerce and therefore in the environment, and it isn't feasible for the EPA to regulate all of them. So we need a scientific way to decide which chemicals are going to have a thorough risk evaluation. Even when we don't know the real life exposures or toxicity, we can still make a good decision by looking at range of potential exposures and comparing them to the range of potential toxicities. I have a little stoplight coloration here. So a lower risk chemical is when the range of potential exposures doesn't reach the range of potential toxicities. A moderate risk chemical would be where there's a chance they could overlap or the uncertainty is extremely high, and a higher risk chemical is when the expected exposures overlap the potential toxicities. So this is kind of a weird question. What data will help me find my average water? Representative samples were developed using the National Water Quality Portal. So this is a wonderful resource that has data from EPA, USGS, the states. I created a pipeline to keep only the most relevant samples for me, only surface water, no blanks. And you can see that I wanted to keep the most chemicals possible to make estimates. And some of my data cleaning reduced the number of chemicals, but the most constraining step was actually the number of chemicals that had at least 50 discrete values. In this context, by a discrete value, I mean a measured value like 3 micrograms per liter instead of a limit value, such as less than 2 micrograms per liter. So this is a highly censored set, as most people who work with water data may be familiar with. So how will I get representative values from this data? I took the mean of the log normal maximum likelihood estimate. As I alluded to before, most chemicals are over 80% censored. You can see the results in this set for 1,4-dioxane. The aqua histogram bars are the count of records that were censored, and the orange are the observed values. And this curve here is my estimated distribution using maximum likelihood estimate. We use the mean instead of the median, which is sometimes used with log normal, because it incorporates knowledge from all of the values, not just the central or most common values. So we're trying to be on the conservative side here with our exposure estimates. Do different sample types change the average? So there are millions and millions of records incorporated in this data set. They were all from different sampling activities. One of the differences we wanted to look at in the set was whether the concentrations from bulk samples were statistically different from those from dissolved samples. You can see my little image here trying to describe what I mean by bulk and dissolved samples. I used text analysis on the description of the methods to categorize the samples into bulk, suspended, or dissolved. And hopefully none of them look like this red one in the back, right? So this color bar here shows the log octanol water partition coefficient, and that basically means the affinity of a chemical for organic material versus water. So we would expect there to be higher concentrations in the bulk samples than the dissolved samples for those chemicals. But as you can see, we didn't really see that. This dashed line is a one-to-one correlation, and most of the means fell along that. These ones, very low ones in the lower left corner, these are so close to zero that I'm not going to worry about those. So we know in real life that this does matter, taking a bulk sample or a dissolved sample. So I think this just reveals some of the uncertainty from using so many of these different samples and taking a mean. So are my representative ranges informative enough for risk prioritization? Remember, when they're too uncertain, we can't make an assumption or a valid prioritization. It's better than an assumption. So the representative ranges do overlap hazard concentrations in Daphnia for some chemicals. So Daphnia is a ubiquitous freshwater species. It's also very sensitive. So again, we're trying to make a very conservative look here. These black dots are the predicted no effect concentrations for Daphnia in our chemicals. And the vertical lines are the expected, the estimated representative ranges. So if you remember the stoplight coloration from before, for most chemicals we're in the green, but there are some chemicals for which all the expected concentrations exceed the toxicity values. The interesting thing about this approach is you can see that none of the most toxic chemicals are actually the highest priority because they're just not showing up in the environment so much. So this is the chemical prioritization schema that we wanted to do, and we were able to do it with this set and this method. So thanks for the opportunity to present, and thanks for listening, and I look forward to any questions you have.
Awesome, thank you. We have 2 minutes for questions. So if someone has one, either put it in the chat or raise your hand on the reactions bar. Susan White.
You know, I always have questions. This is great. This is great. Thanks for kicking us off. I'm wondering where you're going to go next with this.
This is only a small part of my research. We can only make estimates for chemicals that are well sampled, as you can imagine. So we have some nation scale fugacity models developed by EPA, Health Canada, United Nations Environmental Program, and we're calibrating this fugacity models against what we've seen in real life in the United States and seeing if we can make any sort of estimates that would be even more uncertain, but about chemicals for which there are no observations based on their physicochemical properties.
Thank you.
Okay, good start, good start. I'm going to transition us now to the next one. So let's see who we got next. We've got Zhenzhen Zhang from NC State University, and she is going to be telling us about nature-based schoolyards and how they enhance students' perceptions of schoolyard benefits. So Zhenzhen, take it away.
Thank you. We just saw many great posters from elementary school kids. And today my research is also focused on children and schoolyards. The public school system is an important stakeholder to adopt nature-based solution and expand nature benefit to children and community. Compared to traditional school grounds covered by concrete or open space covered by turf and few trees, green schoolyards with natural features such as gardens and woodland are more likely to provide benefit to students and communities. Green schoolyards as the bridge to connect children and nature can provide a variety of benefits to students themselves. For example, schoolyards can provide recreational and educational opportunities and also can enhance students' school experience and also support physical and mental health to children. In addition, the wide range of ecosystem services associated with green schoolyards, such as stormwater management function, provide food source, and support a wildlife habitat, may benefit broader communities. Along with the greening schoolyards process, as students are the main stakeholder in schools, understanding their perception about the benefits of schoolyards and the use of different schoolyards components can ensure schools deliver a wide range of benefits to children. However, little research explores students' perception of schoolyards benefits. Therefore, we began to address these research needs by analyzing how does the presence and the use of natural features shape students' perception of schoolyard's benefit. So we hypothesized that students' perception of benefits to self and benefits to community would be predicted by the presence of nature-based attributes and the use of schoolyards for nature-based activities by students and teachers. So in our study, we surveyed 199 third to sixth grade students from Wake County about their perception of schoolyard's benefit to self. For example, we asked the benefit to self, how do you agree with a schoolyard is a good place to learn? Do you think your schoolyard is attractive? Do you think your schoolyards help you feel calm? And we also asked the student the benefit to the communities. For example, we asked, do you think your schoolyards help make the water cleaner, help reduce the flooding, and help provide wildlife habitat? So students would give 1 to 5 score according to how much they agree on these different statements. We also ask students about the frequency of nature-based activities, such as the play with plants, play with insects or small animals, and play outdoor activities led by teachers. At the same time, we also measured the nature-based activities. At the same time, we also measured the nature-based attributes, including the gardens and woodland existing in the schoolyards. And we found that children were more likely to identify benefits to self than benefits to community. For example, we found that recreational and learning benefits were mostly notified, whereas the benefits to community, like the making water cleaner and helping reducing flooding, were least identified benefits. So when we analyzing how nature-based attributes and the use affect the perception of schoolyards benefits to self, we found that the presence of gardens and woodland were positively associated with students' perception of schoolyards as a good place to learn. And we also found that students spending time outdoor for class activities also positively associated with the learning benefit. In addition, we found looking at or playing with the plants is also positively related, but the relationship is a little bit weak. For the benefit, for students think the schoolyard is attractive, we found that looking at or playing with insects or small animals and spending time outdoor for class activities is positively associated with attraction benefits. And looking at or playing with plants is a little bit weaker, positively associated with the attraction. And we also found that looking at or playing with plants is positively associated with the benefits that students will think schoolyards will help them feel calm. Regarding to the benefit to community, we found looking at or playing with small animals and teacher-led outdoor activities to be positively associated with the benefits of schoolyards to support wildlife habitat, but the relationship was a little bit weak. We also found a weak positively relationship between teacher-led outdoor class and students agree that schoolyards help reduce the flooding issues. So overall, we found that students view green schoolyards in a really positive way. Students' positive view of schoolyards are more influenced by the nature-based activities and teacher-led activities rather than the greening schoolyards alone. As the public school systems are the promising partners to expand the nature-based solutions and expand the nature benefits, we suggest that schoolyards greening efforts should be paired with policies and curricular practice to encourage interaction with nature-based attributes and nature-based elements to ensure that all the benefits associated with the green schoolyards can be noticed and identified by the students. Thank you.
Excellent. Thank you very much. Time for one question. If anyone has one, raise your hand or put it in the chat. Thank you, Zhenzhen. If you have chats that come up or ideas later for questions, put them in the chat. The students will be here the rest of the conference because they're so excited about possibly winning the competition and or attending our event later. So with that, we will move to Haley Plaas from UNC Chapel Hill, and she's going to chat with us about cyanobacteria growth and microcystin production on the Chowan River. Haley, take it away.
All right, thank you, John. Hi, everyone. My name is Haley Plaas. I'm a second-year PhD student at UNC's Institute of Marine Sciences, and today I'm here to talk to you about my research on the aerosolization of harmful cyanobacterial blooms, their toxins in their cells. So jumping right into the background of this, a harmful cyanobacterial or blue-green algae bloom is an issue threatening water quality and public health in our freshwater and estuarine ecosystems across North Carolina and globally. And human activity is providing for the expansions of these blooms, which are linked to the production of toxins associated with various diseases. And one of those toxins, which I primarily study, is microcystin, and it is a potent liver toxin. So we know quite a bit about the health risk associated with the ingestion of microcystin via contaminated drinking water or food, but we know much less about the impacts associated with its inhalation. But we do have plenty of evidence to suggest that these cells and dissolved toxins can be found in spray aerosols. And the mechanism that causes these aquatic cells and toxins to go airborne is called aerosolization, and it occurs at the sea-air interface via bubble bursting. So wave action and wind action entraps air bubbles beneath the water surface, and as those rise through the water column, they can collect all sorts of dissolved chemicals and particles that are suspended in the water. And when those bubbles reach the surface and burst, they eject water, salts, and then all of the chemical and biological components that are also found in the water. So my research is interested in characterizing those spray aerosols, which form during cyanobacterial bloom conditions in the environment. And so where we're doing this in North Carolina is along the Chowan River in Albemarle Sound, which has endured historical nuisance cyanobacterial growth, and they have reemerged in the past decade. So the two main groups of cyanobacteria that we've seen blooming here in the past couple of years have been Anabina or Dolichospermum and Microcystis. And microcystis is associated with really high microcystin concentrations. So the Dolichospermum is associated with lower concentrations of microcystin, but it is also known to produce this toxin. So in 2019, we actually had concentrations measured up to 620 micrograms per liter, which is more than 60 times the EPA's recommendations for swimming. And that was linked to this enormous microcystin bloom during the late summer. So due to heavy cyanobacterial activity in this area in years back to back, and also the sizable fetch in the Albemarle Sound, allowing for greater wave action, this is a perfect area for us to study the incorporation of these cells and toxins into aerosol to investigate it as a potential public health threat. So this past summer in 2020, we ran a field campaign, and the first aim of our study was to survey the concentration of cyanobacterial DNA and microcystin in spray aerosol. And then our second aim was to assess any environmental controls on the aerosolization of those toxins. And we collected water samples in parallel for comparison in collaboration with a group of community scientists from the Chowan-Edenton Environmental Group. So in order to carry out our objectives, we deployed these aerosol samplers on loan to us from the EPA at 2 field stations. And our campaign ran from June to September throughout the peak cyanobacterial bloom season. And these impactors work essentially as a giant vacuum. They collect aerosols onto filters, and we collected both single day and 2-week integrated aerosol samples. And then we also measured real-time ambient mass concentrations of aerosol, PM 2.5 specifically, or aerosol with 2.5 microns or below in diameter. And we're using those real-time particle concentrations scaled with some of our meteorological conditions, such as wind speed and direction, to estimate the relative spray aerosol production. And our water samples were collected on filters or in aliquots, and we're analyzing them for DNA, microcystin concentration, algal pigments, cell counts, nutrients, etc. So jumping right into my findings, our initial scan of those aerosol samples did not reveal any detectable microcystin. However, we're currently in the process of getting those rerun because our HPLC crashed in the middle of that run and our internal standard was messed up. So we can't say with precise confidence that we didn't quantify any microcystin just yet. However, in integrating those peaks, it did not appear as if there was any quantifiable microcystin, but this was not unexpected given the low concentration of microcystin measured in all of our water samples for the summer. So all of the microcystin that we did detect was below 1 microgram per liter, which is considered a win for water quality. But if you do look at the microcystin data that we did collect from our water, the data points that we do have show that the most abundant forms of microcystin were MCLR and MCLA, or with amino acid side chains, leucine alanine and leucine arginine. So these forms are actually much more likely to be enriched in aerosol due to their hydrophobicity compared to some other more benign congeners of microcystin. And so although our toxin production was low for the summer, there was still quite a bit of cyanobacteria bloom activity, even though it wasn't microcystis, that strain that's really associated with the heavy microcystin production. So we're still analyzing our DNA and aerosol with collaborators at NC State. And while I don't have any findings to share with you just yet, I can tell you that we used 16S rRNA primers for all bacteria and cyanobacteria, as well as microcystin encoding genes. We're using a shotgun analysis to look at DNA there. And we're moving forward with a method where we finally were able to get some yields from our aerosol filters. Extracting from quartz fiber filter wasn't working out as well as we wanted it to. So hopefully we have some findings on that soon. And then actually moving into next summer and looking at our plan for this upcoming season, we have some improved instrumentation that we're going to be using to collect microbial life in aerosol. So we're going to be using some of these biospot samplers, which allow us to collect aerosol directly into water. So with that, we'll be able to concentrate our samples much more and also culture some of our findings. And we won't be using those PM 2.5 cutoffs moving forward because we might be excluding some of the larger cells that could be found in aerosol. So basically this last summer was a pilot study for us, and we've improved our methods and we're moving forward looking to next summer, hoping to capture more toxic conditions, even though we're not wishing that on water quality in that region. With that, that's basically a little crash course of my research from the last summer. So thank you all for listening, and thank you to all of my collaborators who helped me get to this point.
Thank you, Haley. We have 1 minute left for any questions. So all you HABS people out there, pepper her with something.
Don, we have a raised hand from Michael Paul.
Excellent. Michael Paul, you're up.
Hey, Haley, I have a question for you. Just, has anyone ever looked in prop wash or boat wakes to see if there's aerosolization that occurs behind a boat where people might be, you know, water skiing or kneeboarding or something?
Yeah, that's a great question. That's definitely something that's been recognized as a potential source of spray aerosol, but I don't think it's actually been quantified. At least I haven't come across anything that's quantified the aerosol production from that, or any sort of recreational activity like with jet skis or something similar.
And then we have one more in the chat before I get you off the hot seat, and it's from Astrid, and she wants to know, what could be the potential concentration factor due to lung filtration? And maybe even small concentrations are a concern.
Yeah, so I could talk a while about the toxicological impacts associated with concentrations that we are seeing in aerosol. The microcystin concentration that has been measured in aerosol is along the magnitude of about 10 nanograms per cubic meter. And so I don't think that there's any sort of association with an acute response to this. I think that in the studies that have looked at the impact of microcystin in the mice of lungs, they're not seeing effects anywhere below like 3 milligrams per kilogram of body weight per day. So there's quite a big dichotomy there between the toxic effects you would see and what the environmental concentrations are. So I think the big question is whether or not there are chronic impacts from this, the environmental concentrations.
Excellent. Thank you, Haley. We are going to move now to Jackie Hartman from UNC Charlotte. And Jackie is going to be telling us about the effects of stream restoration on macroinvertebrate communities in urban development areas. So Jackie, take over the screen and impress us.
Right. Minimize this. Okay. Oh, shoot. Sorry. Okay. Hi, everyone. My name is Jackie Hartman. I'm a second-year master's student in the Geography and Earth Science Department at UNC Charlotte under the advising of Dr. Sandra Clinton. And today I'll be discussing the effect of stream restoration on macroenvironment communities across urban development and agricultural land use changes. So the study region for this project is in Mecklenburg County within the headwaters of the Reedy Creek watershed. Overall, the entire Reedy Creek watershed is roughly 14 square miles. It is mostly forested with some neighboring land uses being predominantly residential due to the fact it is partially protected in a nature preserve. And here the topography is mostly gentle rolling hills with the elevation ranging from 650 to 850 feet. And the watershed was deemed impaired due to a lack of diversity of aquatic insects, and there was significant amounts of erosion. So as I said, our study sites are in the headwaters of the overall Reedy Creek watershed. These streams highlighted in red up here are those that were restored. And so today I'll just be focusing on 3 of our overall 11 stream site locations. I'll be looking at, let me see, our D1 site right here. So our D1 site located right here, and that is where all of this developed region drains right into that stream. And then we have a agricultural subwatershed in which all of this will drain into our A1 location. And then we have a C2 site in our control region where C2 actually was not restored during the restoration. So here's just a quick visual of before and after restoration D1 location in which all that urban development area drains right into the stream. So we can see before restoration, it's really incised, highly eroded, and due to that often water reaching the stream faster due to all the urban development, it was really channelized and really straight. So after restoration, now we can see the stream pattern is more sinuous, so reflect more of the natural stream. They also were able to effectively raise the stream bed about 5 to 10 feet to reconnect it to the associated floodplain. And we can see they were able to plant some riparian vegetation on the stream bank as a suitable habitat for macroinvertebrates. And here's a unique aerial photo that shows just how sinuous they were able to make that stream. It was in March 2018. And we can see here now it's really curved and no longer really straight and narrow. And that also how close it is to this neighborhood that is draining into the headwaters right here that will later go downstream to the D1 location that I'll be talking about. So for the purposes of today's analysis that's included is an average of the seasonal pre-restoration data and then an average of the seasonal post-restoration data. And I'll be looking at the mayfly, stonefly, caddisfly, EPT richness, and then also the NC biotic index, which is an index that is reflective of the unique tolerance value that's assigned to each species, with a lower value indicating that that taxa is really sensitive to pollutants and contaminants, and a higher value is that taxa is really tolerant of stressors in the environment, and then how abundant the taxa is, if it's rare, common, or really abundant, and that will reflect an index indicating we can classify it. If it is lower, the water quality is deemed as more excellent, and if it is higher, the water quality is deemed as more poor. So here you can see pre-restoration, we can looking at the average overall EPT richness, and then also our other overall taxa richness, and that other taxa is just the ones that are more tolerant, so our Diptera, Coleoptera, Megaloptera, those taxa, but we can see that D1 and A1 are having similar patterns in that there's lower EPT richness in comparison to our control that wasn't restored. And there's a greater other taxa present that is more tolerant. And that is reflecting in the NCBI score as it is actually higher, deeming the water quality is more fair in D1 and A1. And with our control region that has more of that EPT taxa present, the NCBI score is actually lower. And it's showing, deeming that the water quality is greater in comparison to D1 and A1. And then after restoration, it's still kind of a similar pattern for our D1 and A1 site. Unfortunately, we do see that the EPT taxa has dropped after restoration, and then that other taxa is becoming more abundant. But it's important to note that those taxa are tolerant to those stressors and disturbance, and restoration is a disturbance. So it can take time for these EPT taxa to perhaps come back and recover properly after restoration. But we're seeing that there is a decrease in that richness. And again, the NCBI is increasing, reflecting that in comparison to our control, the water quality is deemed as more poor in comparison to our control region. And here is our EPT richness and NCBI value trends over time. So there is going to be some seasonal variation because we do collect our samples seasonally. But overall, we can see that after restoration, overall the EPT richness has decreased and the NCBI values have therefore increased after restoration. So overall, the urban development and agricultural influence after restoration had a decrease in EPT richness and an increase in NCBI values. There was no evident change in our forested control site, but overall, these results are suggesting that our land use and the pre-restoration condition could impact the trajectory of the macroinvertebrate community recovery. And it's important to acknowledge it can take many years to see an outcome of success in a restoration project. We roughly have about 2 years post-restoration data, but still we could need more to accurately assess the outcome of this project. But also important to note, in the future, we're going to be looking at all of the 11 sites and also incorporating our seasonal analysis to determine the impact of collecting seasonally on these results. And so thank you. Now I'll be accepting any questions.
Thank you, Jackie. Well done. The floor is open. We have a couple minutes if anyone would like to ask Jackie about her work that she presented. And Michael Paul has put in the text, Jackie, how long do you expect it may take these streams to recover? And then there's a second part. Are there ways for adults to easily get into the restored reaches?
It's hard to say with, I've seen like multiple articles that say, it could take 10 years to accurately assess if these streams have been an outcome, but it's just more to look at the trends over time. And like, again, like I said, we only looked at 2 regular sites that were restored and then 1 that wasn't restored. So I would suggest, I think adding a few more years of post-restoration data would make a really big impact on the results.
All right, any other questions before we let Jackie off the hot seat? Oh, Barbara has a question for you. Have you done any work to quantify various habitat presence in the channels, i.e. riffles?
So it is a riffle pore morphology at Reedy Creek, but the way our collection methods is more qualitative. So to assess the general taxa present at the location, and then based upon what taxa present, we're able to quantify the abundance that way. But I haven't looked specifically at whether there's a difference in the riffles or in the pools in the stream.
Okay, and Jackie, there's one more in the text that you can answer after we get rolling with the next presentation, because we've got to keep rolling. These are lightning talks, but there's a couple more for you in the chat, Jackie. Thank you. And so next I will call to the stage Nancy Lee Alexander, and Nancy's coming to us from NC State University, and she's going to tell us about assessment and reporting of North Carolina private well water quality. Nancy Lee, take it away.
Thanks, John. Yeah, so I'm Nancy Lee Alexander. I'm a third year PhD student in the environmental engineering department at NC State. And I'm excited to talk to you guys about some of my work with private wells. So why are we interested in studying private wells? Well, in North Carolina, approximately 30% of residents rely on private wells as their primary source of drinking water. And while this is a relatively large percentage of residents, private wells fall outside the purview of the Safe Drinking Water Act and don't have really any required regular testing. If residents do wish to have their wells tested, the available testing can be expensive, and oftentimes the results that participants or residents will get back can be a bit confusing. In private wells, we're interested in what's in them because they can be contaminated from various sources. Some of these sources include septic systems, the actual makeup of the aquifer that the water is drawn from, agriculture, or even the plumbing that's used to distribute the water inside of the home. While not everything that we find in private well water obviously is harmful, it is important for us to understand what private well users are drinking and using. So our study, one of the primary objectives is to aid in filling the knowledge gaps surrounding private wells. And to do this, we're hoping to sample 150 private wells in 5 different counties in North Carolina and develop occurrence data for volatile organic compounds, metals and metalloids, anions, coliform bacteria, and pesticides. And to do this, we've split our project into 3 primary phases. And first, recruiting and sampling private wells. Second is analyzing those samples and the associated data. And finally, a big part of our project is communicating those reports or results back to participants and other stakeholders. So as a part of phase 1 of our study, we mailed out flyers which contained project information to private well owners in 3 different counties in North Carolina. If residents received our flyer and wished to participate, they contacted us and we set up a sampling time. And this sampling actually took place from August to November of 2019. So at each location, in addition to actual water samples, we also collected well characteristic data and demographic data using a questionnaire. So the water samples that we took from each well were analyzed by 2 different labs. So at Virginia Tech, we had our samples analyzed for metals and metalloids using an ICP-MS. And the remaining samples were analyzed in our environmental engineering lab at NC State. And these samples were analyzed for VOCs using a GCMS. We analyzed them for coliforms using IDEX quantitrays. We analyzed them for anions using an ion chromatograph. And then finally, we analyzed them for pesticides using a non-targeted analytical method on an LC-QTOF. So traditionally, pesticides are looked for in well water using targeted methods, which use low resolution mass spectrometry or LRMS. And the key to these types of methods is that researchers or investigators really know what they're interested in looking for. But as we know with the contaminants of emerging concern and kind of a lot of these organic contaminants, the list just keeps growing. So these methods can't really keep up with the growing list. So we actually chose to use high resolution mass spectrometry or HRMS to develop a non-targeted analytical method. And really what this does is it lets us kind of collect information on unknown features that may be in our sample. So things that we might not even know we need to look for. So as you can imagine, the data sets for these types of analyses are quite large and we're still going through them. So we're not ready to report those results back at this time. The remaining analyses we did for VOCs, metals, anions, and coliform bacteria were targeted, and these had much smaller data sets. So we have been able to compare these to the EPA maximum contaminant level, action level, maximum contaminant level goal, and the secondary maximum contaminant level to get an idea of if our analytes occurred at potentially unsafe or elevated levels. And on the graph on the right here shows 5 different groups that we split our analytes into based on where they fell in comparison to each of these standards, action levels, or health goals. In the y-axis, you can see the number of analytes that fell into each of these categories. So the main takeaway from this is that we had 51 analytes that we never detected in any well, while we only had about 14 analytes that occurred at potentially unsafe or elevated levels. So while there were only 14 analytes that we detected at potentially unsafe levels, they did occur in a lot of the wells that we tested. So again, you can see 3 of these groups and the percentage of wells that fell into each of those groups. And this 96% of wells did have at least one contaminant that occurred above the MCLG. And we really think that this, the driver behind this is these 4 different analytes on the right. So coliform bacteria, uranium, arsenic, and lead. These 4 contaminants each have MCLG values of 0, and we did find them in a relatively large number of wells that we tested. So if these were detected at all, they fell above the MCLG. So we think this is why this percentage is so high. Another thing we were interested in is looking at the impact that plumbing materials may have on the water quality. So to do this, we took 2 samples. We took a pre-flush sample, which was taken as soon as we turned on the kitchen tap. And this is most likely representing what homeowners or residents would be using or drinking. And then we took a post-flush sample, which was taken after running the tap water for 5 minutes. And this most likely represents the water that's actually coming from the well. So as you can see, there's a pretty large difference between these 2 samples. And this is the total average concentration of metals associated with plumbing. So we actually saw that we could decrease the average concentration by about 650 parts per billion, or 60%, if we ran the water for 5 minutes before taking the sample. And this is really important for homeowners or residents because they could potentially reduce their exposure. So as I mentioned, we are reporting all of these results back to our study participants. And to do this, we have partnered with Silent Spring Institute, and we'll be giving participants their results through an online digital exposure report back interface, or DRRI, as you can see here. So their reports will be, they can access them through an online portal, and they are interactive. So they can kind of click through all of here, read anything, or as much or as little as they want, but they can find individual analyte results. They can compare their results with other study participants' results. And then we'll also give them some treatment recommendations based on what their results contain. Yeah, so thanks for listening, and I'm happy to answer any questions.
Excellent presentation. Thank you, Nancy Lee. Anybody have a question for Nancy Lee? Giving people plenty of time to type. All right, you must have explained it so perfectly. There are no questions, Nancy Lee. Thank you very much. Oh, there we go. Andrew George wants to know, which counties did you sample?
Yeah, so right now we've only sampled Wake, Guilford, and Ashe County. But we're hoping to sample some more, yeah.
There you go. All right, thank you, Nancy Lee. We're right on time. Nancy Lee, there's still some stuff coming into the chat. You can keep an eye on that. But we will move now to Leah Weaver. She's also from NC State University, and she's going to be talking to us about fungal bioremediation of neonicotinoids. She wanted me to have to say that word. Good job. I appreciate it. In water, a potential low-cost treatment. So, Leah, take it away.
All right. Hi, everyone. My name is Leah Weaver. I'm a second-year PhD student at NC State, working under Dr. Tariq Aziz in the environmental engineering program, just like Nancy Lee. My work focuses on using fungi to remove neonicotinoids, which are a type of pesticide, from water systems. And we're particularly interested in this because we believe it has the potential to be a good, low cost, low maintenance treatment, hopefully, for certain applications. And before I get going, I just want to say thank you to WRRI for funding this work. We're just getting started though. So today I'll be sharing some background, the questions we're trying to answer, and our plans to answer those questions, but I don't have any results to share with you today. So in this context, fungal bioremediation is referring to white rot fungi, which are a group of fungi that have been shown to degrade a whole host of emerging contaminants. The fungus I'm interested in particularly is Phanerochaete chrysosporium, which has been shown to degrade neonicotinoids as well as other contaminants. And the way it's able to do that, as well as other white rot fungi, is through this whole host of enzymatic systems that the fungi produce. So in this case, lignin and manganese peroxidases and cytochrome P450s, which in the fungus's natural environment, these are used to degrade lignin in the woody biomass that the fungus is growing on, but they're also able to co-metabolically degrade these sorts of contaminants. Now, while neonicotinoids have been degraded, so we know this technology works, this particular group of contaminants has not been closely studied. So we're really interested in understanding which of these enzymes are most important, how much they contribute, and then also looking at some more non-traditional sort of systems. So we're interested in growing the fungus on a woody biomass so that you use that biomass to support the fungus's growth without having to add like a glucose solution or something like that. And we think this has a good potential for applications for more on-site type treatments, especially with stormwater runoff, maybe on the edge of an agricultural field, something like that, where a low maintenance technology could be especially useful. Now, neonicotinoids are a group of pesticides. The three we're interested in are imidacloprid, clothianidin, and thiamethoxam. And these three make up about 25% of pesticides used globally. So they're very widely used both around the world, in the United States, and here in North Carolina. And part of that is because they have both agricultural and residential uses. Now, imidacloprid specifically is a residential compound. And so you can see in this map where imidacloprid has been tested in surface waters, that is especially prevalent in urban areas. But I have not been able to find evidence or data collected for clothianidin and thiamethoxam. So it's probably there because these compounds are so widely used, but it has not been tested for. Now, these concentrations found in North Carolina are at a relevant concentration. So these compounds are toxic to invertebrates. That's kind of their intended use. They're meant to be pesticides, but they also are toxic to non-target organisms like aquatic invertebrates. And so this concentration of about greater than 0.1 microgram per liter has been shown to have chronic effects for aquatic ecosystems. And these compounds have also been found in treated drinking water and wastewater elsewhere in the United States. So we know that these compounds are recalcitrant to traditional treatment. We need to be thinking about alternative ways to remove them from both surface waters and treated waters. So the question we're really interested in asking is, how can we best design a fungal bioreactor to treat for these contaminants? We need to understand what the kinetics are for the fungus growth, enzyme production degradation, how these different enzymes are contributing to the degradation, what different pathways are being used, and then ultimately, how can we optimize that degradation? And all of this under realistic conditions. If we were to put this out in a field, what sort of conditions would be available as far as oxygen, as far as retention time, these sorts of things that may not have been considered previously in a more controlled system, or what we're interested in looking at. So the way we're going to approach this is we'll be doing some degradation experiments, looking at how many enzymes are produced, under what circumstances, how the neonicotinoids degrade under these different circumstances. And then our goal is to take the information we gain from that and apply it to an agent-based model that we've developed to help us optimize design without having to do everything in the lab. So the goal is to understand what's important, maybe how much biomass we need and that sort of thing, model it on the computer, and then eventually build a system in the lab to test those optimal conditions for degradation. We're also interested in the future and doing some work on the transformation products that these fungi are creating from the neonicotinoids, and also looking at, in a non-sterile system, what sort of community is present, how might that impact the system, and hopefully we'll get some symbiosis involved to improve our degradation even further. So as I mentioned, we really believe that this has a potential to be a cost-effective treatment. And these are the reasons why. So we're hoping that we can use a waste product like a wood chip or sawdust, something, maybe corn cobs, as a carbon source for the fungi. So you're not having to go in and feed them glucose and ammonia all the time. They can sort of subsist. And because this process is co-metabolic, as long as the fungi are happy and growing, they're also able to degrade these contaminants at the same time. You don't have to train them to do it. You don't have to have a certain concentration. It's just happening simultaneously. And if we are able to grow them in this way, it should be relatively low maintenance. And then we're also hoping to do this in a non-sterile environment, and so explore what community is present and how different community members might contribute to support this degradation. So that's the gist of what we're working on, what we're hoping to do in the future, and I'd be happy to answer any questions.
Okay, thank you, Leah. If folks have questions for Leah, you know the routine by now. Let's get them in the chat or raise your hand.
So Leah, this is Susan. Who do you feel are going to be the adopters of this?
Yeah, so there's definitely some work already being done for wastewater treatments in like an on-site sense, so like hospitals and that kind of thing that have high concentration of contaminants. But we're really interested in exploring this for stormwater. So maybe something like a stormwater management, or on-site in like an agricultural application where you're applying these pesticides and might be having them in runoff.
And Leah, there's a question in the chat wanting to know if you see these fungi being implemented in stormwater ponds.
Yeah, so I'm not an expert in stormwater management yet. I'm not exactly sure what specific infrastructure might be already existing where they could be applied, but definitely having potential for like urban stormwater management applications and first flush applications.
And then a couple more. How long do fungi take to grow before you can see results?
Yeah, so that's one of the things we want to look at is exactly how much time do you need? How long can the fungi be sustained before, you know, they're too old and they don't work? But right now it's looking at like 5 to 7 days. These fungi grow pretty fast. So hopefully that won't be an issue for implementation. And
then one final question for you. What are you going to do with the fungi once they have been contaminated?
Yeah, so the idea is that the fungi are able to degrade the contaminant. They're not uptaking it and holding on to it. And so hopefully, if we're able to look at transformation products, we would see that the neonicotinoids are being degraded into something less harmful. So the fungi should be able to be disposed of in a more normal way without having to worry about hazardous materials and that sort of thing. They're not a pathogenic fungi or anything like that either. So.
Excellent. Well, thank you, Leah. And I'd like to give all our students a virtual applause for their great efforts. WRRI really does feel our ability to let students give presentations at this conference is one of our calling cards. And so we're happy we were able to do that in this virtual environment. And so now the judges are going to go quickly confab and come up with a winner. It's going to be a hard job. And in the meantime, I will turn it to Anna, who will provide some closing remarks and next steps. Anna?
Yeah, thank you, John. First of all, I just want to say thanks to everyone who is still on the line and joining us. We've had a really jam-packed agenda today. I want to thank all our panelists, as well as our students, You know, we've worked really hard over the last several months to put this together. There's definitely been challenges. We've done a few shorter virtual events, but this is our first kind of foray into trying to condense the annual conference down to, you know, something that's digestible. So we really appreciate your time and your attention and your just planning to be with us today. We have another wonderful session put together for tomorrow, and we realize it's Friday, so that's a little tough sometimes. But we do hope you will come back and join us. We have plenty of space, so even if you didn't sign up or you have a friend who wants to join, please do spread the word. If you can't make it back tomorrow, that's okay too, we understand. We've got some links on the bottom of the agenda, and you can also find these on our website. We have a survey online that you can give us your feedback. Please just take that once. If you're going to be here tomorrow, you can save it to the very end and let us know how we can improve. We want to get better. We want to know what we're doing right. So your feedback is real valuable to us. And then I know a lot of folks are here for the PDH credits and the CEUs. So we've got some instructions online how to receive those as well, but do feel free to reach out with any questions. Again, our sessions today and tomorrow have been recorded. We are going to be trimming these up and putting some closed caption with them, and they will eventually be available on our website. Maybe not next week, but soon. I want to invite you all in a few minutes after John announces our student winner. We're going to take another short break that is not on our agenda listed, but we'll take 5 minutes. But we're going to go into a networking session. So we have 23 career panelists that have signed up to stick around. Many of them you heard speak today. But yeah, we want to invite all of our students, anyone who has questions or just wants to follow up a little more. We have a document that was shared with you, our networking kind of menu du jour, so you can figure out which room to get into. So we're gonna, like I said, we're gonna, oh, John, I see you are back. Are you ready to make an announcement?
I am back. Thank you for giving the judges a chance to collate all our great information. And I'm pleased to announce that Jackie Hartman from UNC Charlotte has won today's competition. So congratulations, Jackie. And we will follow up with you later about getting you all the great accolades and rewards that come with this honor. So congratulations.
Thank you very much. Thank you.
Yeah, I'll echo John's congratulations, Jackie. We appreciate your time and attention, and invite our audience to tune in tomorrow. We have another great set of students. We have 7 presentations tomorrow, so you'll hear a whole new version of talks. So we are about to enter the networking portion of our event, and we thank you for everyone who has again volunteered your time for this, and we hope you have some really good conversations. So in just a few minutes, our technician, Rob Chapman, is going to launch the breakout rooms. And these are labeled according to the networking menu that Susan, I believe, shared a link to our website. So you should be able to find that there as a Google document and also as a PDF available for anyone who wants to download. And I will stay here. This is called our main session. So if you guys want to jump back and forth between rooms and come back and see what's going on, feel free to do so. And if I don't see you again, we hope to see you tomorrow afternoon.